presenters. NF-kappa-B pathway is extremely important in stimulating proliferation and survival of CLL cells in the niche. As you see on the right side, NF-kappa-B pathway, along with the B-cell receptor signaling pathway, were the two most upregulated pathways in the lymph nodes. The truth of the matter is, even though B-cell receptor signaling, where a lot of our targeted therapies currently act, does induce NF-kappa-B pathway, it is also induced through a multitude of other mechanisms in CLL. So there are two main components to the NF-kappa-B pathway. One is so-called canonical, which is upregulated in the presence of B-cell receptor signaling and CD40 receptor signaling. The other one is non-canonical, and CD40 receptor signaling also stimulates that. Additional signals which upregulate NF-kappa-B include BAF, APRIL, toll-like receptor, and others. It's a cascade of events. In the canonical pathway, an IKK complex forms and then promotes ubiquitination of I-kappa-B alpha. That results in release of P50 and P65 effector subunits and translocation to the nucleus. In the non-canonical pathway, there is a P100 precursor protein which undergoes proteasome-assisted degradation to then form P52, which similarly translocates to the nucleus and drives transcription of cell cycle proteins, pro-survival proteins such as BCL2, as well as several chemokines. So both of these pathways require presence of the calling ring ligases, such as E3 ligase. E3 ligase is activated through a multi-step process, which essentially requires the NED8 activating enzyme. So the interaction of the moiety called NED8 and NED8 activating enzyme is what MLN4924 targets. MLN4924 forms a covalent adduct with NED8. And that prevents activation of NED8 activating enzyme and therefore all subsequent steps. As a result, E3 ligase is no longer active, and therefore ubiquitination of either I-kappa-B alpha or proteasomal degradation of P100 cannot occur. As a result, those effector subunits cannot translocate to the nucleus. So we modeled the lymph node niche by using a well-established model which employs CD40 ligand expressing cells. As reported previously, in those cells, when they are co-cultured, in CLL cells, when co-cultured with CD40 ligand expressing stroma, there is activation of the NF-kappa-B pathway. Here we show non-canonical pathway, increased processing of P100 to P52, increased expression of MCL1, BCLXL, and stable expression of BCL2. I'll show the activation of canonical pathway later. Predictably, this kind of stroma reverses resistance to most known chemotherapy agents, including fludarabine, chlorambucil, bendamustine, as well as novel agent CAL101. So interestingly, when cells were cultured in the presence of MLN4924 in this type of system, resistance did not occur. So MLN4924 not only fully reversed stromal-mediated protection from spontaneous apoptosis, but also induced apoptosis in excess of what occurs off stroma. So we analyzed what MLN4924 was doing there, and indeed, consistent with the mechanism of action, we saw a reduction of NED-elated kalins, which occurred very early on. Along with that, we saw accumulation of phosphorylated I-kappa-B alpha. And if you remember the previous slide, that we would predict that will be accompanied by reduced translocation of effector NF-kappa-B proteins. There are other targets for the calling ring ligases, including CDT1, P27, P21, and we looked at several of these, and here's an example of P27, which doesn't change in this system. So MLN4924 actually reversed activation of NF-kappa-B, and here we show the non-canonical NF-kappa-B activation, where you see disappearance of P52 subunit, which occurs fairly early, after 8 hours of incubation with the drug. Then there is accumulation of phospho I-kappa-B. If you look at this, when you look at this figure, In a control sample without the drug, all the p65 canonical effector is located in the nucleus, where the nucleus is yellow here, superimposed over blue DAPI. When you add MLN4924, you can appreciate, hopefully, the semilunar shapes which correspond to translocation of this p65 subunit to the cytoplasm. And same happens with p52. The nucleus is yellow in a control sample, and then upon treatment with MLN4924, there is translocation of this subunit back into the cytoplasm, resulting in those semilunar shapes. So we ran a microarray. Since this drug also can target other proteins besides I-kappa B alpha, we ran a microarray to determine whether NF-kappa B is certainly is affected. And of the top 4 pathways affected by the drug and CLL cells, were all they were all NF-kappa B related pathways one way or another. There was a reduction in cytokine synthesis, including CD40 itself. There was a reduction in BCL2, BCL-XL synthesis, and an increase in BEAM. So importantly, we found that a well-recognized target of NF-kappa B, coded by BIC gene, which is MIR155, was one of the most reduced targets in our gene expression array. So we confirmed, using a transcription factor assay, we confirmed that this effect was dose-dependent. And here you see a decrease in P60 and P65 and P50 DNA binding. And then we validated several of our genes and again saw a dose dependence in MLN4924 effect. So when we went to protein expression, however, we didn't see that much of a change in BCL2 expression or BCL-XL expression, likely indicating the reasonably long half-lives of those proteins. However, we did see an increase in BEAM expression. And the jury is still out whether it occurs due to reduced degradation of BEAM, as at least one isoform, which is extra long BEAM isoform, can be assisted by a 3 ligase, its degradation is, or whether it's increased transcription. But we did see increased transcription of BEAM upon introduction of MLN4924. And then recent data indicated that NOXA degradation is dependent on kalindrin ligases, and we did see an increase in NOXA upon incubation with NED8 targeting drug. So knocking down BEAM and NOXA partially abrogated the pro-apoptotic effect of NED8 targeting drug. However, just for control here, knockdown of PUMA did not have any effect. So then we asked the question, since we are upregulating BEAM, somehow BCL2 is still there. So what happens if we combine MLN4924 with a BH3 mimetic, which directly targets BCL2? So that would be ABT199, just mentioned in by the previous speaker. And indeed, while CD40 ligand expressing stroma induced significant protection of CLL cells from toxicity induced by ABT199, in combination with MLN4924, that was fully reversed, and we saw a rapid induction of death in the presence of both drugs. Thus, targeting NED8 presents itself presents a novel mechanism of toxicity in CLL cells. It synergizes or cooperates rather with several agents, including B-cell receptor targeting agents, which I didn't show here. And there are certainly other mechanisms involved in the mechanism of action of MLN4924 in CLL cells, and we are investigating this further at this time.